In our last video, you met food number 1 on the autophagy stack, the compound nature cell biology, confirmed in August 2024 is the single missing key your recycling engine cannot run without. Today, I want to answer what that video did not have time to cover. Exactly how much do you need? Which specific type of aged cheese? And when do you eat it to keep your autophagy running every hour of every day, with no fasting required? Because right now, you may be eating aged cheese every week and receiving almost none of its biological benefit. Not because the science is wrong, because the dose is wrong, the type is wrong, and the timing is wrong. By the end of this video, none of that will be true for you. Stay until minute 9. The cell timeline I will show you will permanently change how you see your refrigerator. For decades, the conversation around autophagy told you one thing: fast longer, fast harder. The more you restrict, the more your cells clean themselves. And that advice is not entirely wrong. But it is incomplete in a way that costs you every hour you spend fasting, because it missed the most critical variable in the entire equation. In August 2024, a research team published a landmark study in Nature Cell Biology titled "Spermidine is Essential for Fasting-Mediated Autophagy and Longevity." What they discovered changes everything we thought we understood about how fasting actually works. When researchers blocked spermidine synthesis inside the cells of their subjects, fasting stopped working. Not partially, completely. The autophagy benefits disappeared. The longevity markers reversed. The cellular cleanup crew did not arrive. Their conclusion was clear: fasting does not directly activate autophagy. Fasting triggers the release of a specific polyamine compound inside your cells. That compound activates the autophagy program. Spermidine is not a byproduct of fasting. It is the mechanism your fast depends on entirely. And here is where the science gets precise. Spermidine does not work through mTOR suppression. It does not work through AMPK activation. It works through a completely separate epigenetic mechanism, operating at the level of your gene expression itself. Layer 1. It inhibits an enzyme called EP300, releasing 3 critical autophagy genes simultaneously. Layer 2. It activates a translation factor through a process called hypusination, building new cellular recycling infrastructure from scratch. Layer 3. It does all of this independently of whether you have eaten, whether your insulin is elevated, and whether mTOR is active or suppressed. But here is the problem: that mechanism only activates above a specific biological threshold. And almost nobody eating aged cheese right now is actually hitting it. That is exactly what the next section is about. Here is why almost every conversation about spermidine misses the only question that actually matters for your body: how much? The mechanism does not activate gradually. It activates above a biological threshold. Below that threshold, the EP300 enzyme remains active, the 3 autophagy genes stay locked, and the recycling machinery stays dormant. You can consume spermidine every single day and receive no measurable benefit if the signal never reaches the level required to unlock the program. A clinical study conducted at a research center in Berlin confirmed that 6 milligrams of spermidine per day is the minimum dose at which measurable autophagy flux is detectable in human immune cells. 6 milligrams. That is the number. Above it, the mechanism fires. Below it, it does not. The typical daily diet delivers between 1 and 3 milligrams of spermidine. Most people eating without specific intention are permanently below threshold. They are consuming the compound. They are not triggering the mechanism. Now, which cheese closes that gap most efficiently? Not all aged cheese is equal. Spermidine concentration varies significantly with aging time, microbial culture, and the protein breakdown process called proteolysis, which produces spermidine as a natural byproduct of the aging process itself. Aged Parmesan and aged cheddar contain among the highest spermidine concentrations of any commonly available food. Well-aged varieties reach up to 20 milligrams per 100 grams. At that concentration, 30 grams, roughly the size of your thumb, delivers approximately 6 milligrams of spermidine, the exact threshold your cells require. Aged Gouda and Brie fall in the range of 10 to 13 milligrams per 100 grams. You would need close to 50 grams to reach the same threshold. That is still achievable, but it increases your saturated fat and sodium intake considerably, which is worth considering for daily use. Blue cheese and soft ripened varieties are inconsistent. Spermidine content varies widely by batch, milk source, and production method. They're not a reliable daily source for hitting threshold. Processed cheese, pre-sliced formats, and shelf-stable varieties contain negligible spermidine. Heat treatment and emulsification destroy polyamine content almost entirely. These are the appearance of a source, not a source. For those asking about wheat germ, it contains 24.3 milligrams per 100 grams, the highest of any commonly available plant-based food. 25 grams delivers the full threshold dose. If you cannot tolerate dairy or prefer a plant-based protocol, wheat germ is your most effective alternative. For those asking about supplements, clinical trials have tested 1 to 10 milligrams per day from standardized extracts, with the strongest human autophagy data at 6 milligrams. Supplements are a practical option when your diet cannot consistently reach threshold. But food-derived spermidine arrives alongside spermine and putrescine, two synergistic polyamines that appear to enhance the overall biological effect. The food source delivers the complete signal. The supplement delivers one part of it. The next question is one almost nobody asks, and it is the one that determines whether any of this actually works for you. The aged cheese on your supermarket shelf today may be biologically inert. Here is exactly how to tell the difference. The most important thing I can tell you about buying aged cheese for autophagy is this: the label is not the evidence. The biology inside the cheese is the evidence. And most cheese sold as aged in standard supermarkets has gone through processes that make the label accurate and the spermidine content almost meaningless. Here is why. Spermidine accumulates in aged cheese through a process called proteolysis. As the cheese ages, naturally present enzymes break down the casein proteins in the milk. That breakdown process, over months and years, produces spermidine as a direct byproduct. The longer and more completely that proteolysis occurs, the higher the spermidine concentration. Heat treatment, emulsification, and accelerated processing interrupt that chain at every stage. Processed cheese, pre-sliced formats, and shelf-stable varieties contain spermidine levels so low that studies have measured them in the range of less than 2 milligrams per kilogram. That is not a source. That is background noise. So, how do you identify the real thing at a physical store? First, aging time. You need a minimum of 12 months of continuous cold aging. 24 months is significantly better. Parmigiano Reggiano aged 24 months or more carries a protected designation of origin status, which legally controls the production process and eliminates the use of synthetic additives and genetically modified inputs. That designation is printed on the rind. It is verifiable. It is your first confirmation. Second, the crystals. Inside a well-aged Parmesan, you will see small white granules embedded in the paste. These are tyrosine crystals, and they form as a direct result of prolonged enzymatic activity during aging. The same enzymatic process that produces tyrosine crystals is the process that generates spermidine. If the crystals are present, the proteolysis occurred. If they are absent in a cheese labeled aged, the aging was insufficient or interrupted. Third, the taste. A properly aged hard cheese delivers a sharp, peppery, slightly bitter bite at the back of your throat. That intensity is proteolysis made sensory. If the cheese tastes mild, smooth, and neutral, the aging process did not go far enough. You are tasting low spermidine content. Refrigerated storage at the point of sale is a baseline requirement. Any aged cheese stored at room temperature on an open shelf has already begun losing polyamine integrity. Now you can identify the right cheese. The next question is when to eat it, and what happens inside your cells hour by hour when you do. Now follow spermidine from your plate to your cells in real time. Within 1 to 2 hours of consuming your 30 grams of aged Parmesan, spermidine appears in your bloodstream. Studies measuring plasma concentration after oral consumption confirm detectable levels within that window. The compound absorbs through your small intestine, enters circulation, and begins moving toward every cell in your body. The delivery phase is complete. Your cells are receiving the signal. Between hours 2 and 6, spermidine reaches your cell nucleus. EP300 inhibition begins. The 3 autophagy genes, ATG5, ATG7, and ATG12, release from suppression simultaneously. Autophagosome membrane nucleation initiates. LC3 conversion, the direct scientific measurement of active autophagy, begins to rise. Your cellular recycling machinery is not waiting for a fasting signal. It is already building. Between hours 6 and 12, autophagy flux is fully active. Damaged proteins and dysfunctional organelles are being engulfed, processed, and recycled. This is the cleanup window. This is what most people believe only fasting can produce. You have produced it with food. From day 1 through day 7, consistent daily consumption keeps spermidine elevated in your blood and tissues. LC3 remains measurably elevated when dietary intake stays at threshold. The signal is no longer a spike. It is a sustained state. From day 14 to day 30, the EIF5A hypusination pathway drives measurable TFEB upregulation. Your cells are no longer just running the existing recycling program. They're building new lysosomes and new autophagosomes. The recycling infrastructure itself is expanding. This is not maintenance. This is a cellular upgrade. Now, here is how your complete 8-food protocol integrates with everything you have just learned. Your morning coffee suppresses MTOR through chlorogenic acid. Your olive oil builds TFEB from outside the nucleus. Berberine and quercetin fire AMPK from 2 directions simultaneously. And your 30 grams of Parmesan reaches the epigenetic layer that none of those compounds can access. Each mechanism covers a direction the others cannot. Together, they leave autophagy with no biological reason to stay off. Before you build this protocol, 3 categories of people need a specific warning first. Stay with me for 60 more seconds. Before you build this protocol, 3 specific groups of people need to hear this first. First, if you regularly experience headaches, skin flushing, rapid heartbeat, or digestive discomfort after eating fermented or aged foods, you may have a reduced ability to break down histamine. Aged cheese is high in histamine. In that case, aged Parmesan may be a better choice than Gruyere or blue cheese. And starting with a smaller portion is the right approach while you assess your response. Second, if you are managing high blood pressure, 30 grams of aged Parmesan contains approximately 390 milligrams of sodium. That is within a safe daily range for most people, but it requires tracking within your overall daily sodium intake. Monitor your numbers and adjust accordingly. Third, and most importantly, if you are currently taking MAO inhibitor medications, do not add aged cheese to your daily diet without speaking to your doctor first. Aged cheese contains tyramine, and the combination with MAO inhibitors can cause a sudden and dangerous increase in blood pressure. This is not a minor caution. This is a direct contraindication. Please take it seriously. For everyone else, screenshot the protocol on your screen right now. This is your complete daily autophagy stack. Every food, every timing, every mechanism covered. Now comment cheese stack below and tell me one thing. Which cheese are you adding to your protocol starting today? Then click the video on your screen for the complete fasting protocol that combines with everything you just learned.